Jesus was asked what it will be like, in the last days, when he returns.
As it was in the days of Noah, so it will be also in the days of the Son of Man. Luke chapter 17 verse 26.
So, what was it like in the days of Noah? There was wickedness throughout an unbelieving world. God warned them of his coming judgment but was ignored. Then the flood came as promised. Genesis chapter 6 verses 5 to 7.
Then the Lord saw that the wickedness of man was great in the earth, and that every intent of the thoughts of his heart was only evil continually. And the Lord was sorry that he had made man on the earth, and he was grieved in his heart. So the Lord said, I will destroy man whom I created from the face of the earth, both man and beast, creeping thing and birds of the air, for I am sorry that I have made them.
There was an interesting situation regarding the state of humanity just before the flood. Human DNA was being corrupted by fallen angels, who had offspring with women. Genesis chapter 6 verses 1 to 2 and 4.
Now it came to pass, when men began to multiply on the face of the earth, and daughters were born to them, that the sons of God saw the daughters of men, that they were beautiful. And they took wives for themselves of all whom they chose.
Sons of God are fallen angels.
When the sons of God came into the daughters of men, and they bore children to them. Those were the mighty men who were of old, men of renown.
The fallen angels and the human DNA created very intelligent mighty men.
To further make this point, God says, Noah was a just man, perfect in his generations. Noah walked with God.
Perfect in his generations does not mean he was a morally perfect human, but that he was genetically perfect in his lineage. God had to save the integrity of human DNA to protect the lineage of Jesus Christ. And where are we today? The discovery in 1953 of the double helix, the twisted ladder structure of deoxyribonucleic acid, DNA, by James Watson and Francis Crick, marked a milestone in the history of science and gave rise to modern molecular biology, which is largely concerned with understanding how genes control the chemical processes within cells. Rosalind Franklin was an English chemist and X-ray crystallographer whose work was central to the understanding of the molecular structures of DNA. The Human Genome Project was an international scientific research project with the goal of determining the base pairs that make up human DNA, and of identifying, mapping and sequencing all of the genes of the human genome from both a physical and a functional standpoint. The original cost to read a single human genome was $100 million. Today you can read it for $250. The discovery of the CRISPR-Cas9 as a genome editing tool represents the work of many scientists from around the world. In 2012, scientists were able to use the CRISPR-Cas9 system as a cut-and-paste tool to modify genomes.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then, they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA-cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
CRISPR-Cas9 has given scientists the ability to change human DNA. It is cheap and easy to use. There are scientists all over the world using this new system of gene editing.
CRISPR gene editing technology is a tool that scientists can use to change the letters of DNA in cells in precise ways. So I like to use the analogy of a word processor on our computer. So we have a document, you can think about the DNA in a cell like the text of a document that has the instructions to tell the cell how to grow and divide and become a brain cell or a liver cell or develop into an entire organism. And just like in a document, the CRISPR technology gives scientists a way to go in and edit the letters of DNA, just like we might cut and paste text in our document or replace whole sentences, even whole paragraphs or chapters. We can now do that using the CRISPR technology in the DNA of cells. And over the last few years, as this technology has begun to be deployed globally in for different applications, I've found that I have gone from thinking about it initially just with sort of almost wide-eyed excitement, thinking about all the opportunities that this offers, to realizing that there was real risk, and that we really needed, we meaning the scientific community and really frankly the human community, needed to be aware of this and discussing it. And one of the things that sort of brought that to the forefront of my mind was a dream that I had fairly early on, in which I was, I walked into a room and a colleague of mine said to me, Jennifer, I'd like you to explain the CRISPR technology to a friend. And he brought me into a room, and a person was sitting with their back to me. And as they turned around, I realized with sort of a horror that it was Hitler, and it was actually Hitler with a sort of a pig nose, and it almost looked like a chimeric pig-human sort of creature. And it was it sounds funny in a way to relate that image, but in the dream it was a terrifying thing, and I really felt real, just stone cold fear in the dream, and sort of woke up from that dream with a start and realized, kind of had this initial feeling of what have I done, and
But these are not the typical silkworms.
This one right here, one of the parents tested out at full-strength spider silk. So I have great hopes, and they just hatched this morning.
Did you catch that? Spider silk? Keep watching.
We're taking the black widow gene and putting it in place of the silkworm silk gene. And so when they go to make the cocoons, they read the gene and they just make it. And instead of making regular silk, they make spider silk.
At first glance, these cats look like any normal cat, but there are two big things which make them very different. One, the cats are clones. Two, they glow in the dark. You heard right. When they're put underneath ultraviolet light, they glow a dull red. And it's no accident. Scientists manipulated the donor cat's genetic code, then passed those changes onto the clones.
외부 유전자를 도입한
It's meaningful as we introduce outside genes to transgenic cloned cats for the first time, and as they have the red fluorescent protein gene in their organs, they give off a red color.
Scientists in Massachusetts are growing replacement limbs, and they're starting with rat legs. They're calling them bio-limbs. They have blood cells, tendons, muscles, everything. An electric current could even make them flex. So how'd they do all that? They flushed out the cells of one rat so it's just bone. Then they dumped cells from a second rat into that hollow leg, and the cells begin to grow. Now get this, the leg is made up of the second rat's cells so the organ doesn't get rejected. When you flesh out the organ, it turns clear like this. Donor cells in this heart were flushed out and replaced with the cells of another animal. So we've started putting human cells into monkey arms.
Photos of extremely muscular pigs bred by a farm in Cambodia have been doing the rounds online, sparking controversy about genetically modified animals. The Hulk-like pigs are apparently bred by Durak, Cambodia, a pig farm in the Asian country's Banteay Meanchey province. The company's Facebook page is littered with photos and videos of these freakishly muscular animals that appear to be the result of some extreme genetic tampering. Some have accused the breeder of using hormones and steroids to turn the pigs into nightmarish monsters. But seeing as Durak, Cambodia is also selling insemination kits to people wanting to grow their own, their unnatural physique is most likely caused by genetic modifications.
Officials at the National Institute for Agricultural Research in France are currently investigating a major breach. A genetically modified research lamb bred to have the DNA of a jellyfish was sold to a slaughterhouse amongst a group of normal, unchanged livestock. It was then sold to a buyer in the Paris metropolitan region in October 2014 and is thought to have been consumed.
Humans have been drinking it for millennia. Wholesome, healthy, full cream milk. But this dairy in Beijing isn't quite what it seems. These cows have all been genetically modified, and this is human breast milk. The Chinese scientists responsible successfully inserted the human breast milk gene into a cow embryo and then implanted it into a surrogate. The result is a transgenic herd of 300, and milk the scientists claim is healthier than the bovine variety.
Yuval Noah Harari gave keynote speeches on the future of humanity in Davos in 2020 and 2018, on the World Economic Forum's main Congress Hall stage. He regularly discusses global issues with heads of state. What would some elites do with this incredible ability?
Many tyrants and governments wanted to do it, but nobody understood biology well enough, and nobody had enough computing power and data to hack millions of people. Neither the Gestapo nor the KGB could do it. But soon, at least some corporations and governments will be able to systematically hack all the people. We humans should get used to the idea that we are no longer mysterious souls. We are now hackable animals. Data might enable human elites to do something even more radical than just build digital dictatorships. By hacking organisms, elites may gain the power to re-engineer the future of life itself. So does the data about my DNA, my brain, my body, my life, does it belong to me, or to some corporation, or to the government, or perhaps to the human collective? Humans are now hackable animals. You know, the whole idea that humans have, you know, they have this soul or spirit and they have free will and nobody knows what's happening inside me. So whatever I choose, whether in the election or whether in the supermarket, this is my free will, that's over. Free will, that's over. That's over. Over.
Are we in the last days, as in the days of Noah? Thank you for watching. We pray you have eyes to see what God is showing you.